Here are two genetically identical twins, Jackie and Jill, but Jackie may be more susceptible to a disease that is highly based on genetics. How come? That's where epigenetics comes in, the study of how factors other than our genetic code affect our gene expression. But first, what are histones? Histones are proteins that pieces of DNA can wrap around, just like this piece of yarn wrapped around this pencil. DNA is negatively charged, and histones are positively charged, and just like in love, opposites attract. The histones that DNA is wrapped around consist of 8 main histones and 1 linker histone. There are many types of modifications that all function with their own special enzymes that take them to the histone. Modifications can turn the DNA on and off, and even help in DNA repair, all without editing the genetic code. In acetylation, the HAT enzyme brings an acetyl group to a lysine residue on the N-terminal tail, canceling the charge of the histone. This leads to the DNA being more loosely wound, accessible for transcription, essentially turning the DNA on. Deacetylation reverses this process, turning DNA off. Deacetylation can play a really big role in cancer. Cancer is the uncontrolled growth of tumor cells, and genes like p53 can help suppress them. However, in cancer, a lot of the times genes like p53 are overly deacetylated or overly turned off. That's why scientists are experimenting with HDAC inhibitors, which bring levels of deacetylation down to normal for tumor-suppressing genes. This can lead to tumor suppression. What's incredible is that histone modification patterns can be inherited, but they can also be affected by our environment. For example, exposure to toxins like lead and arsenic can affect immune function. But arsenic isn't toxic, it's medicinal, and it's really pretty in green pigment. P.S. Arsenic isn't medicinal or pretty, it's really toxic, even in green pigment. But green's my favorite color.